Hello and welcome to Bosons Botany. I'm starting with kinds of DNA, or basically, I'm starting with DNA, the topic DNA, which is very important. So, there are many kinds of DNA A, B, C, D, E, but recently zDNA is discovered. So, zDNA is a very recent discovery and it's very common in eukaryotic organisms, but bDNA is the most common type of DNA that we find in almost every plant and every animals including human beings. So we're gonna study both B and Z DNA and will differentiate some important points to classify both B and Z DNA. So that is something kept for the future video. So here is the second point. All these have minor differences in structure with respect to number of base pairs in a turn or coil or spiral, in diameter, length of one spiral, phosphate orientation, etc. So there are some minor differences in the structural view of point. If you take into consideration the number of base pairs in a particular turn or coil or spiral, even differences in diameters, length of one spiral, then phosphate orientation, etc. So by observing these differences, you can categorize whether a DNA is A, B, C, D, E or Z DNA. So here is the chemical structure of bDNA. As I have already told you, bDNA is the most important type of DNA which is found in every animal and plants and human beings. So we're gonna study this in detail. So there are two strands known as polynucleotide chains. And why polynucleotide? Because there are many nucleotides forming a polynucleotide chain, and these nucleotides form the building blocks. Now, if I distinguish nucleoside and nucleotide, then it will be very easy for you to know what are nucleotides exactly and how they are produced. So nucleoside, first nucleoside. Nucleosides are produced when sugar and nitrogenous bases combine. So here are four types of nucleosides which are present in DNA. These are adenine or deoxyriboadenosine. Here, if you observe this, this is the ribose deoxyribose sugar that I have already told you in my previous video. So this is the sugar portion, this is the ribose sugar, ribose pentose sugar, and on carbon number 1, this adenine nitrogenous base is attached. Adenine belongs to purine nitrogenous base, or purine has adenine that I have already told you, and it consists of two rings. One is 5-membered ring and the other one is benzene, or not benzene but 6-carbon ring. So this adenine nitrogenous base is attached to the first carbon of ribose pentose sugar of DNA, and if you number this is 1, 2, 3, 4 and 5. So attached to carbon number 1 of pentose sugar, the nitrogenous base, to form deoxyribose adenosine. This is the first nucleoside. The second nucleoside here is of guanosine, and same deoxyriboguanosine. And here, as you know, that both adenine and guanosine belong to purine base pair or purine nitrogenous base. So exactly like that of adenine, here also guanosine or deoxyriboguanosine is formed by the attachment of guanosine on the first carbon of deoxyribose pentose sugar, which is very clearly visible from this diagram. The third type of nucleoside is deoxyribocytidine, and here cytosine, as you know, belongs to pyrimidine base pair having only one 6 carbon ring. So this is also attached to the first carbon of ribose pentose sugar. And the final type is deoxyribothiamidine. This also belongs to pyrimidine, which is attached to the carbon number 1 of ribose pentose sugar. So nucleosides, these are composed of sugar and bases, and here are 4 important nucleosides, which are deoxyriboadenosine, deoxyriboguanosine, deoxyribocytidine, and deoxyribothiamidine. Now if I come to nucleotides, these are nucleoside plus phosphate. Very simple. When you attach the phosphate group with nucleoside, you get nucleotide. And I have not drawn all these structures here, but just to give you an idea about nucleotide, here I have drawn only for adenine. So just like nucleoside, just attach adenine to carbon number 1, then number the carbons on the ribose pentose sugar like 1, 2, 3, 4, 5, and on carbon number 3, here is the attached phosphate group, and this is the phosphodiester linkage. This red line here, this is the phosphodiester linkage, and this is formed by elimination of water molecule that I have shown here. One water molecule is being eliminated during the formation of phosphodiester linkage. So on carbon number 3 of the sugar structure, we have the phosphate group, which forms the phosphodiester linkage. So this overall structure is nucleotide for adenine, and we will name this nucleotide like this: deoxyriboguanosine. Sorry. Okay, sorry for that little break. So for adenine, the nucleotide is deoxyriboadenosine monophosphate. So just write monophosphate against nucleoside. Just like here, deoxyriboadenosine for nucleoside in case of adenine. For nucleotide, you will just write monophosphate. So deoxyriboadenosine monophosphate or adenylic acid. This is the nucleotide for adenine, where we have phosphate group attached to the carbon number 3 having a phosphodiester linkage with the elimination of a water molecule. So similarly, deoxyriboguanosine monophosphate or guanylic acid, deoxyribocytidine monophosphate or cytidylic acid, and deoxyribothiamidine monophosphate or thymidyllic acid. So I think this is pretty clear, and the concept is well understood by you guys. Just pause the video, go back and just examine all the structures that I have drawn and the names that I have written to make sure you understand the difference between nucleoside and nucleotide. Why I have not written or included uracil here? Because uracil does not belong to DNA. It is only for RNA. So I have not written uracil here because it is not a part of DNA. So yeah, just have a look on both these nucleosides and nucleotides. So at 3' position or at 3 carbon position of deoxyribose sugar, the phosphate group is attached. Also, at 5 carbon position of sugar, we have the phosphate group. Therefore, the direction of DNA strand is in the 3' to 5' direction, or vice versa. This will become clear after having a look on the diagram that I have drawn down below. This direction in one strand will be upward, while in the other strand it will be downward. So keeping these points in mind, just go down and see the diagrams. I have drawn the chemical representation of DNA, which is extremely important to understand the core structure of DNA. So this is a DNA having two polynucleotide strands. If you further enlarge the diagram, you will get to see the phosphate groups here. These are the phosphate groups. S stands for sugar, or the deoxyribose sugar attached with the nitrogenous bases like adenine, having double bond or 2 hydrogen bonds with thiamine. Guanine has 3 hydrogen bonds with cytosine, and thiamine has 2 hydrogen bonds with adenine. Now here comes the actual diagram of DNA. So you might get very confused. Let me just explain this. Okay, this is the whole structure of a DNA, or the overview of a DNA, showing its chemical structure. So if you see here very systematically, I will explain you. This is the sugar structure, or the deoxyribose sugar. I have drawn 3 such sugars here, one after the other, like this. Now number every single carbon present on this sugar structure, starting from carbon number 1, 2, 3, 4 and 5. So before I explain you this diagram, let me just mention one thing here. Carbon number 5 is nothing but it is CH2OH. Okay, so just keep this in mind. This is CH2OH. An alcoholic group is present. Okay, so these are sugar that I have drawn here, sugars. Number them like I have said. Now attach any nitrogenous base. It could be anything. I have attached guanine here. So guanine attached to carbon number 1 like this. Then onto the second sugar we have attached adenine, and on the last sugar we have attached cytosine. Just randomly you can select any nitrogenous base you want. I have drawn them randomly without any strict restrictions. So just draw the sugars on carbon number 1. Now on carbon number 3 you need to draw the phosphodiester bond, and for that you need to attach phosphate groups. So here I have drawn the phosphodiester bond like this. So if you remember, phosphoric acid or phosphate, it is H3PO4 or OH, double bond OH. So This is the structure of phosphate group or phosphoric acid, not phosphate but phosphoric acid. But if you remove two hydrogens from here like this, then two negative charge will come on oxygen and we will have H3PO4 minus 2. And this is the actual group that we attach on carbon number 3 and also carbon number 5. So this is the phosphate group, H3PO4 minus 2. So on carbon number 3 we have attached one of the oxygen like this to give phosphodiester linkage. Similarly, one of the oxygen which has a negative charge on this, if you just recall this structure here, one of the oxygen having negative charge will bond with the CH2 group with the elimination of water, that is H2O will remove. Similarly, here also on carbon number 3 a phosphate group is attached like this, showing a phosphodiester linkage, and here we have the OH group. So once again, sugar. On carbon number 1 we have our nitrogenous base. On carbon number 3 we have the phosphate group showing phosphodiester linkage, and on carbon number 3 we have the OH group, and on carbon number 5 we have CH2OH. So this is 3' to 5' direction. If you number the carbon atoms present on sugar, then it will be easier for you to give the direction. So it is 3' to 5' direction going upward. Now, since there are two nucleotide strands, therefore it is known as polynucleotide DNA. So this will be towards the downward side. So guanine will bond with cytosine having triple bond or 3 hydrogen bonds like this. So just invert the structure that I have already drawn on my left hand side. Just reverse the orientation, and this is the sugar. Number it 1, 2, 3, 4, 5. On the carbon number 1 we have attached cytosine, which is complementary to guanine, that I will discuss in Chargaff's rule later on. But I think you know that guanine will bind with cytosine having 3 hydrogen bonds. Adenine will bind with thiamine having 2 hydrogen bonds. So cytosine bonded with guanine with 3 hydrogen bonds, which is attached to carbon number 1. Then carbon number 2, carbon number 3, we have the OH group here. Carbon number 4, carbon number 5, we had CH2OH, but OH will eliminate, sorry, H2O will eliminate, or you can also say OH will eliminate in the form of water. And here we will attach the phosphate group, just like the previous nucleotide strand that I have shown on my left hand side. Like this, on to our next sugar, we have carbon number 1, which is having thiamine attached to adenine with 2 hydrogen bonds. Then carbon number 2, carbon number 3 having the phosphodiester linkage with phosphate group. Carbon number 4, carbon number 5, we have CH2 attached with phosphate group along with phosphodiester linkage. And this is the last sugar having guanine with 3 hydrogen bonds with cytosine. This is carbon number 2, carbon number 3, phosphate group having phosphodiester linkage, carbon number 4 and carbon number 5. So the orientation which is coming here is 3' to 5', which is downward. So this is the actual chemical representation of DNA. It is not at all difficult. If you practice this um, maybe 5 times, you will master on this, trust me. So just remember this diagram, just practice it on your own without any help, and just try to understand the orientation, the position of carbons, and you will be able to understand and you are good to go. So just take a look, pause the video. This is very simple. So this is the 3 prime 5 prime direction, and this is 3 prime 5 prime direction, or you can see 3 to 5 prime and 5 to 3 prime, whatever you want to say. Only anti-parallel direction is there in DNA. Just remember this, anti-parallel orientation is followed in DNA. So I think this is clear. So I'm winding up today's video, and after that I will give you the DNA double helical structure and comparison between bDNA and zDNA. So there's lot to know, lot to understand. Just revise what I have taught and practice this diagram. It is very important. Thank you.